Our next speaker will be introduced by James McKenna from the Department of Anthropology.
Well, yet again, I have the honor of introducing Dr. Alison Fleming, who has been at the University of Toronto, many of you know, for about 36 years. And I suppose if there was an Academy Award to get for academic excellence, indeed, our friend here has received it. She is one of the very few Canadian research chairs in neurobiology and a fellow in the Royal Society of Canada. And I noticed, Alison, on your website that you maintain that you're not fixated on any particular paradigm, and your scores and scores of wonderful students that have populated and educated us certainly attest to that. She's asked proximate questions and evolutionary questions. She studied the young, the old. There probably isn't anything on the neurobiology of parenting that Alison hasn't touched. Okay, there you go. So I introduce you, and please welcome Alison, who will be speaking on caregiver responsivity, prompt responses to needs.
I have to move my mic. Three of my wonderful students are actually sitting, they drove from Toronto. They didn't really warn me that they were going to do that. I sort of knew, but... Okay. I changed my title a little bit to Dopamine, another magic bullet in the regulation of caregiver responsivity. And this is work that has been going on for quite a long time, both at the University of Toronto and at McMaster in Hamilton. But before I start, I want to thank Thank you for inviting me. This is a great conference, and it really is quite exciting, and I'm really glad my students get to listen to every perspective. They get the narrow mechanistic perspective from me, and here they're getting the more broad, clinical, and applied perspective as well. So I'm really pleased that we can now see the connections. Okay. The work that we've been doing, as I said, occurs at Arundel College, or UTM. It was Arundel when I first arrived in 1975, as well as St. Joseph's Hospital. I just want you to see, sometimes people show pictures of southern France, or beautiful pictures of field stations. Well, we are in a city, and our buildings are all very old. So I just wanted you to see that. We studied... My mic's not on. We study... it's too loud. Can you turn it down, please? Because I really hate yelling. Okay. We study, our animal work takes place in this building, and we work with rats predominantly. Our human work takes place in Hamilton. We work with new mothers. We've also worked with new fathers. We work with depressed mothers, and we work with teenage mothers. So we look at a wide variety of different kinds of mothers. The approach that I take really is that maternal behavior is not single, unitary, reflexive. It's not a thing. It's not a structure. Maternal behavior occurs at the time of birth because the mother undergoes many, many changes in her psychology. She undergoes changes in her affective state. She undergoes changes in her how salient or positive the young become for her. She undergoes changes in her attentional abilities. She undergoes changes in aspects of her memory. She undergoes perceptual changes. And all of these changes that occur under, I believe, in part the influence of hormones, as well as other expectations and other psychological factors which predispose these behavioral changes, all contribute to her ability to take care of these young. So we have been looking at changes in multiple psychological propensities and how these are mediated by different brain systems. We believe that there is a maternal system, if you will, a maternal circuitry, but feeding into it are circuitries that are recruited and used by many, many other functional behaviors. There's the emotion system, and there's their perceptual systems, their attentional systems, prefrontal and executive function, prefrontal systems, so that many neuroanatomical systems are involved in the expression of mothering, even though you wouldn't call them part of the maternal circuit. But they feed into the maternal circuit, and I think that's very important to keep in mind. And that also involves many neurotransmitters, and all of this is affected by early experience, by genetics. I'm not actually going to talk a lot about our early experience work. I'll mention it at the end, as well as some very new work we've been doing on the genetics and the G by E and on mothering. Okay, so as I said, today's magic bullet, I'll try to describe how maternal behavior is affected now, not by the oxytocin system, but by the dopamine system. We all develop a preference for one hormone, one neurotransmitter, one behavior, and we fall in love with it, and we talk about it, and I tend to forget what's going on in other worlds, so thank you, Sue. Okay, what I'll talk about today is this part. I'll talk predominantly about the initial response that the mother has to the offspring. And in the case of the rat, that's initial, the first day. In the case of humans, it's the first weeks. But that is, what is it that changes the mother from one that is non-responsive to offspring, perhaps, as we'll see, to one that becomes responsive? How does the mother undergo a change in how salient the pups are, and also how rewarding they become? And I'll make the argument this involves a system, the mesolimbic dopamine system, in part. This ongoing quality of maternal behavior is another area of work that we've been working in very extensively, trying to understand. Once the mother has begun to mother and interact with her young, she engages with those young. Some mothers are sensitive and respond contingently. Other mothers are not very sensitive, don't respond contingently. Mothers have a very wide variety of patterns of responding. I don't think any are better or worse it really depends on what they're being adapted to. And this is maybe something we could have for a discussion, because I really feel that if you go cross-culturally, you find many different styles of mothering, and I think that they are styles which are adaptive given the culture, given the values of the culture. So I'm not actually one person who believes that babies need to be held all the time. I'm in the school of thought that feels that, depending on the culture it's growing up in, that baby needs to be loved, obviously, and cared for, but the actual continual touching, etc., is not something that I think is necessary in some cultures. And I think Heidi Keller is a wonderful anthropologist who's done some magnificent work trying to show the relationship between the child's socialization into its culture, whether it's a culture that values autonomy or it's a culture that values interdependence, and mother's mothering style. And it's some beautiful work, I think. Anyway, this is why I never get to the end of my talks. I just keep chatting away. So there is a whole business about the role of the prefrontal cortex regulating mother's ability to inhibit responses, her attentional systems as well, and that is the ongoing quality of maternal behavior. I'd love to talk to you about it next time. So have another conference. Thank you. Okay, so the dopamine system, it originates in the ventral tegmental area of the midbrain and projects to the nucleus accumbens as well as to the prefrontal cortex. Sorry? Okay. But let me just set the stage for the dopamine system. It turns out that when a female gives birth, and I'll talk both about rats as well as about humans, and I'll keep going back and forth, and see, I try to remind you of who I'm talking about. The signal is that if you see a picture of a rat, I'm talking about a rat. And if you see a picture of a mom here, I'm talking about a mom, a human mom, okay? So in the case of rats, the female rat is actually not very responsive to offspring during when she's not pregnant. In fact, if you give her foster young, she will attack them, she will bury them, she'll stay away from them. She's pretty avoidant. And then if you test her with foster young through the pregnancy, she remains relatively avoidant until the very, very end of pregnancy. There's a shift so that now she becomes much more interested in remaining with the foster pups. And when she gives birth, of course, she's very, very actively involved with them. On her first exposure to young, as primiparous animals, so she will as they come out, they have 16 or 18 if they're in the laboratory, a lot of babies, too many babies to imagine. It's a good thing they don't show much individual recognition. They don't need to. But they will eat the amniotic sac, they will eat the placenta, they'll clean the babies up, and then they'll retrieve them to a nest site, and they will get on them, as we'll see, and then lick them a lot, and then crouch over them. Okay, then what we see, if you look at the endocrine changes that the mother undergoes during pregnancy, and this is very characteristic of many species, it's different in different species in terms of the amounts, but most mammalian species, progesterone is a very, very important hormone. It's the gestational hormone for pregnancy. It maintains the pregnancy, and in the rat, but not in all animals, you begin to see a decline in progesterone about 5-6 days before parturition. Estradiol, another hormone, is relatively low in the rat, although in humans it's quite high during gestation, and it undergoes a rise right at the end of pregnancy, and so you see a shift in this ratio of estrogen to progesterone. This increase in estrogen that you see at the end of pregnancy actually has the function in the rat of putting the female into a state of not only being maternal, but actually also sexual, because rats will mate with males right after birth, and they will in fact be gestating one litter while they're nursing another litter. It's very efficient. If you are a seasonal breeder, you can, the minute you wean one litter, out pops the other. I can't imagine what that must be like. In any case, so we see that the hormonal changes, if given to a virgin female rat who has her ovaries removed, she's ovariectomized, you give her progesterone, you give her estrogen, and then you give her foster pups. She now will become maternal. She'll respond to them in a matter of a couple of days. She'll be very active in response to those foster pups. So we know that those hormones are important. As well, estrogen results in a rise in prolactin, which is another hormone that's been implicated in parental behavior in many species, birds included. This is also a very important hormone. But, of course, there are other hormones, too. Oxytocin, when we've heard a great deal about that, and we know that estrogen affects oxytocin receptors and this hormone. In the rat, this hormone does not act to turn on maternal behavior in the absence of this suite of other hormones behind it. So you have the background of estrogen in particular, and then oxytocin acts in that background. And that's because estrogen actually influences the density or the production of oxytocin receptors. Humans have a different pattern. They also undergo an increase in progesterone. They also have high levels of estrogen during the pregnancy. In general, progesterone and estrogen both decline at the time of parturition. You don't get this prepartum drop of progesterone and rise in estrogen in mothers in general. However, if you actually look at the ratio of estrogen to progesterone across the pregnancy in new mothers, and you divide your mothers up into those that show an increasing ratio of estrogen to progesterone versus those mothers that don't, and then you assess right after birth how the mothers are feeling about babies, their baby in particular, and this is assessed through questionnaires which have been factor analyzed to give us factors related to feelings of nurturance right after birth. What you do find is that those that show an increasing estrogen to progesterone ratio tend to be more positive respond more positively on the nurturant factor. Okay, so that's the background of what happens. Now, we believe these hormonal changes affect mother's maternal behavior by affecting brain systems and neurotransmitters related to all of these psychological variables, but we're going to talk a little bit about stimulus salience and reward today. Okay. So this is the rat. I actually described the behavior. Often I actually have little movies to show what these rats do, but you can see these pups are very, very altricial. They have very thin skins. You can see the milk in their tummies after they've nursed. So they're very immature. Their eyes are closed. Their ears are closed. They're very olfactory creatures, though. They do respond to the mother's odor. They also respond to being touched by the mother. That becomes very important. In response to someone's question about touch, we can have a long discussion later. I think it's really a fascinating topic. But the mother will retrieve the pups to the nest right after birth. She gets in the nest and she starts licking. She licks like crazy. It's really exciting to watch. When I started in this field, all we cared about was retrieval. And that's what I mean. Everyone has their favorite behavior and their favorite hormone. Well, retrieval, that was the name of the game, because animals that don't raise young will not retrieve them. So I saw this as a marker of motivation to mother. But Michael Meany came along, and he shifted my attention to something called licking. You can high lick and you can low lick. You can be in a population. High is good, low is bad. I don't quite agree with that, but nevertheless, licking is very important. We know that the licking behavior has the effect on the pups of altering growth hormone, of changing apoptosis in the brain, of changing levels of dopamine, as well as changing levels of oxytocin receptors. Licking is very, very, very important for normal development. Higher licking in general produces more of these outcomes. So licking is a very important behavior. As well, the mother in licking, she's attracted to the young. She's attracted to the androgen metabolites in the urine. She drinks the urine, and drinking the urine rehydrates her, so when she's nursing, she doesn't have to drink so much water. It's a great symbiotic system. So this licking is like wonderful. But you can have an animal that will lick. You can dissociate the onset of the behavior. An animal will show retrieval responses and then very low licking. But they'll retrieve immediately. So they're motivated to mother, but they're not necessarily doing it at a very high intensity. And I think that dissociation is a very important one we can also talk about later. And then, of course, she crouches over them, and she looks very happy. Okay, so so the question now is, are new mothers very attracted to the odors of babies? We've never been able to show in our lab that mother rats, even tested right after birth, show a preference for pup odors independent of lactation odors. So the odor of the pup is really something that's conferred on those pups by being in a nest with a mother that has milk and whatever else she has. So in these studies, we found that mothers, when tested for their preference for lactating nest odor, and it's either compared to clean nest odor or diestrous nest odor, that's nest odor from a diestrous virgin female, you find that mothers are more attracted to the lactating nest odor at the end of pregnancy, and it really increases right after birth. If you look at non-mothers, you do not get this strong preference. And this has been demonstrated in a number of ways in a number of laboratories now. The shift from avoidance of pups prior to parturition to approach to pups after birth, we believe, results from both the increase in the positive valence of pups, or pup-related cues, and a decrease in fear and withdrawal tendencies. Both must occur for mothers to retain proximity to the young and care for them. In subsequent studies, we gave virgin females progesterone and estrogen, and we then tested them for their maternal behavior, and as has been demonstrated by many, many labs, if you do this manipulation, virgins will become maternal very, very quickly in response to foster pups. They also show a preference for the olfactory, the lactating nest odor, which I find quite interesting. That's independent of having experience with pups. Okay, now we were interested to know whether being attracted to pups also, do the pups constitute a conditioned stimulus for the mother? So once the pups have, the mother is attracted to them, she'll interact with them, she'll lick them, she'll engage with them. Then what happens? Then those pups become actually conditioned stimuli. And the way we showed this, having grown up at Columbia University under the influence of the Skinnerians when I was an undergraduate, we decided to adopt a Skinner box approach to determine whether pups are reinforcing for the mother. And here's a Skinner box. And here are normally animals press bars, and when they press the bar, the food comes into the chute. They eat the food, and then they press the bar, and they eat and you can get continuous schedules of reinforcement going. Well, in this case, our food were little rat pups. And the rat pups were in a little container here in a carousel that every time the female pressed the bar, the carousel would move one step forward, the floor would fall out, the pup would come down the chute, and then the mother could do whatever she wanted. She could neglect them, or she could pick them up and retrieve them. And lo and behold, what she would do before birth is show a very low rate of responding to that bar. This is bar presses per 30 minutes. But this is one animal, once she gave birth, she would show this increase in rate of responding. Now, in the case of food, the rate of responding can be very, very high. But in the case of pups, we can't get that rate very high. It does get pretty high. 30 bar presses per 30 minutes isn't bad. But the reason it's not like hundreds, the way it would be with food, is because the animal has its repertoire of behavior that it has to show in order for the pup to be reinforcing. And what is that? She's got to take that pup, and then she's got to move it over here. And then she's got to start licking it. And then she's got to engage with it. And you're trying to get the pup out of there, so she'll go back and press for another one. You see, so there's a whole sequence of stereotype behaviors that actually control what's reinforcing for that mom. So if you prevent her actually from retrieving the pup, it just drops down. She gets to look at it and smell it and stuff. She extinguishes almost immediately. So that's not adequate. She's got to engage in the behavior. Okay. Among humans as well, new mothers are often very responsive to their infants soon after birth and are attracted to infants and to their cues. This is an example of such a study that we did a number of years ago. We used very high-tech procedures. It's called the Baskin-Robbins study. We take Baskin-Robbins containers. We get different odorants that we put into these Baskin-Robbins containers. So moms are instructed, this is in the hospital on day 2, to put we put their babies on a little nightshirt, okay, and then we cover it up with the other shirt that they normally have on. And they wear that overnight. Then we take those little shirts and we put them in the freezer, okay? And we first rub the baby's head, and then we put it into the freezer. Okay, or we collect urine from the baby's diaper. We have a little thing that we put in there. We also collect the other stuff from the baby's diapers, the meconium or feces later on. We use marjoram. We've used gorgonzola cheese. We've used underarm deodorant, not deodorant, sorry, the underarm other stuff, the axillary stuff, okay? And then we have the mothers they don't know what they're smelling, and they just give us ratings of how attracted they are to this odor. It's very simple. They don't know what they're smelling. If we told them what they were smelling, the demand characteristics would be just too huge, and everyone would be very positive. Well, what you see is new mothers on day 2 postpartum are more attracted to the infant T-shirt than our non-mothers. Okay, this is day 2 postpartum. No one likes urine. No one likes meconium. Mothers don't like the smell of their baby's diapers. People sort of like spice. Now, we've done this with mothers, with fathers, with women at 1 month postpartum. Okay? So, but this is sort of the main point of this, is just that new mothers do seem to be attracted to the odors. Now I'm talking about the odors of their babies. If you divide up how attracted they are by median split, those that are attracted plus 0, 0 plus, those that are not attracted, and you divide them up by median split, and now you look and see you get a number of factors of what's going on in the hospital. What you see that those that are more positive in terms of how attracted are mothers who have their first contact with their babies more rapidly. Okay, that's a latency measure. They nurse their baby, they first nurse their baby sooner. That's a big effect. Okay, and we also observe them in the hospital. We videotape them, and then we record mother-infant interactions. And in this case, we just recorded the amount of time they spent in close proximity, with their noses in close proximity to the baby. They spend more time in close proximity to the baby. Now, the direction of causality here, of course, we don't know. I assume the experience that they engage, they have more time with their babies because they nurse earlier, and they are spending more time with their babies, and that presumably increases their attraction to the baby odors. I think that's the direction of the causality here. And the reason I think that is if you do the same experiment with dads with more experience holding their babies after birth also become more attracted to the odors. Okay? There is a whole literature out there showing that mothers are not only attracted to the odors of babies, they're able to recognize their babies based on their odors. And this is an older literature. Porter and Shaw showed that if moms are presented with their own baby's odors versus another baby's odor, they are able to do that better than chance. So they learn very rapidly about their baby's odors. And also, it turns out, skin surface. They're able to recognize the skin characteristics of their babies, the dorsal surface of the hand, it turns out, more readily than if they've had. They're able to do it more readily than another person is able to do it who is not a new mother. Okay. We've also looked at mothers' responses to the cries of babies. Odors seem to do one thing, and vocalizations do another. And as you'll see when I talk about dopamine, which I hope I'll get to, that really is an important distinction. On self-report mothers, new mothers are more sympathetic and more alerted in response to infant cries than are non-mothers. And these infant cries were played to mothers, and they were played to non-mothers, and they were not cries from their own babies, by the way. Okay? But we found that they were more sympathetic and they were more alerted than were non-mothers. Fathers were too, by the way. But the father had this extra characteristic that he also was more irritated. So sympathy and irritation went together in the father, not in the mother. Okay, what about the role of the brain and dopamine system? As I mentioned to you before, the neuroanatomy is a complicated one. These are just some of the sites that are involved in the neuroanatomy of mothering. We have the medial preoptic area. That is considered, as it were, the final common path. That's the region of the brain which has receptors for estrogen, has receptors for oxytocin, has receptors for prolactin, has receptors for progesterone, estrogen, prolactin. That's pretty good. Dopamine. Okay, so it's a region of the brain. If you lesion it, if you take it out in a rat that is very maternal, that rat will stop responding to pups. You know, it might lie next to them, but it won't retrieve them, and it won't get on top of them unless they push underneath her. Okay, so it's a region of the brain that if you electrically stimulate using a kindling procedure, and they've had a bit of experience. You can't get a virgin to become maternal by stimulating the MPOA electrically, but if the animal has a substrate that's more sensitive, that is, it's had young before, now it would normally not be responding, and now you electrically stimulate it, you can get it to start responding to pups. It's a region of the brain, if you put the hormones in, estrogen in there, you can get an animal to begin responding to the pups. So it's a very important region, and it receives input, as we'll see, from a number of places, and it projects to the brainstem, to the midbrain, etc. Okay. Now, what about this nucleus accumbens, which is the region I'm mostly interested in today? What we see is that this region of the brain is a part of the mesolimbic dopamine system. It receives input from the ventral tegmental area, okay? And it is a system that's been implicated in reward in many, many contexts. Sexual reward, food reward, tickling reward? Huh? Music, everything. The nucleus accumbens is really part of a pathway. Some people call it reward, other calls it reward prediction error, right? Okay, so, pardon?
How about seeking?
And seeking. So it's a system that seems to be involved in
Go get it.
Oh, go get it. I can't say it. It's not just positive. That's been my problem with it, because you think of it as a system that responds only to positive stimuli, but it responds to stimuli that are in fact expected. Okay. Now, the ventral tegmental area has neurons with dopamine in them. They project to the nucleus accumbens. The ventral tegmental area itself receives input from the medial preoptic area. And the ventral tegmental area contains oxytocin receptors. And this is how Sue and I get together, because you see, when there's stimulation from young, it's coming through the medial preoptic area, and all of these things happening in there, and all of these are different neurochemicals and receptors, glutamate receptors and NOS, etc. But the oxytocin cells here project down to the ventral tegmental area. And there are receptors, oxytocin receptors, in the ventral tegmental area. And those receptors, are they on the dopamine neurons? We don't know. They might be on the dopamine neurons, but in any case, those neurons project to the nucleus accumbens. So there's a way for oxytocin to get to the dopamine system. Okay? Okay, what about the nucleus accumbens? If you put into the nucleus accumbens a receptor a dopamine antagonist, and this has been done a number of times in a number of labs, including our own. You put dopamine antagonists into the nucleus accumbens, you will delay the onset of retrieval responses. Animals will not learn about their young, so that you block both maternal memory and you block the initiation of retrieval in these animals, okay? If you put an agonist into the nucleus accumbens in an animal that isn't quite maternal yet, she's a late pregnant animal, she's getting there, but she's not there yet, you put in a dopamine agonist, you enhance the expression of that initial retrieval response. Michael Newman. Okay? So that antagonists block and agonists enhance mothering, the onset of mothering, when put into the nucleus accumbens. We've taken a slightly different tact as well. We want to know what is happening in the nucleus accumbens when the mother is interacting with the young, okay? And so what we did is a very extensive microdialysis study where we measured extracellular dopamine in the nucleus accumbens, and we hope to get up to the cortex. We're not there yet, but at least we have the data for the nucleus accumbens. and we've compared new mothers with non-mothers. We've compared animals that received hormones that turn on maternal behavior versus those that don't. We've looked at animals that have had a little bit of maternal experience and a lot, and we've also looked at animals that are reared without their pups, animals reared without their mothers, excuse me. Okay, so if you compare a postpartum animal with the cycling animals, okay, now this would be on day 4 or 5 postpartum, so they've had experience, okay, they've had some experience, and you just measure the dopamine before you put the pups in, okay? You measure the dopamine through this dialysis probe, and that's assayed through HPLC, by the way. Okay, what you find is the postpartum animal actually has a lower baseline before the pups are put in than do the cycling animals. Lower. So we said lower. It seems to be dopamine is actually being suppressed in some way. Okay? If you now put the pups in and you look at the response to pups in those two groups as a percentage of baseline, the cycling animal doesn't seem to have much of a response to the pups. But the new mother has a big response to the pups. This is in the Nucleus Accumbens, okay? You take the pups out, dopamine drops. This is 8-minute bins, by the way. This difference is reduced if you co-vary the baseline, but it is not eliminated by any means. So you have both a baseline difference and you have a responsivity difference. Is this specific to pups or any stimulus? Well, it seems that the new mother is responsive to pups. In this case, the cycling animal seems to be a bit more responsive to food. So even though if you were to food deprive the mother and food deprive the mother and pup deprive the mother, I don't know what her dopamine system would do or what her response would be, because mothers need food in order to lactate and to nurse appropriately. Okay? But at the very least, it looks like right after birth, mothers are very responsive to the young, and the dopamine system knows it. Okay. She doesn't have to actually interact with them to get that response. But mind you, she's experienced. So she has interacted with them. If you just put pups into a little box here, or food, these are like, what are these, Cheerios, into a little box here, and you look at the percent change in dopamine, the postpartum animal shows a bigger dopamine response just by being exposed to the pups. Okay? In this particular study, we didn't get any food differential. So it does seem to be specific to the pups. Now, we can show that the hormones that turn on maternal behavior are also able to produce this difference in dopamine response to pups. Okay? If you take a virgin animal, you remove her ovaries, you put in the hormones that activate maternal behavior, progesterone and estrogen in this particular case, okay, and now you do the exact same experiment comparing hormone-treated animals with non-hormone-treated animals. This is what you see is an increase in the behavior of the hormone-treated animals and not in the behavior of the non-hormone-treated animals, even though these hormone-treated animals are not yet maternal. So they're going to be maternal. Their system has gotten primed, and they're now, the hormones have primed their system, getting them ready to have that whole consummatory sequence of behaviors occur. Okay? So hormones of pregnancy and parturition affect the dopamine by reducing baseline levels, and hence by increasing the signal-to-noise ratio when the signal, which is the pups, are presented. The dopamine system is therefore primed by hormones and by experience, I didn't talk about that, which functions to enhance the salience of pup stimuli. Now, in terms of humans, it's really hard. We haven't done PET scans. I'd love to do PET scans on new moms, but it hasn't been done to my knowledge. And, but we have looked at, we've begun a series of studies, fMRI studies, And Kat Wunsch back there did the first major study. You can talk to her if you want afterwards. Basically looking at brain activation patterns in new mothers as a function of exposure to pictures of babies. Okay, so here are some of the sites that we looked at, but I'm just going to talk a little bit about the nucleus accumbens actually in the amygdala, but we've actually looked at the entire set of sites. The study has at present 20 subjects in it, 7 primiparous, 13 multiparous. They're 3 to 5 months postpartum. Okay, we get all kinds of information about their mood, anxiety, etc. We also take pictures of their babies. Then a week later, the moms are put into an Fmri, a magnet, where they're presented with baby pictures. We record bold responses to the baby pictures using a procedure that Kat can describe to you later. And then we do home visits and look at their behavior, etc. I won't talk about that. Okay, here are the pictures. So we present them with either their own baby or another baby, same age, same gender. Okay? Most moms can tell who's who, but not all. And it's the not all that I find is going to be the most interesting. Okay? Here the babies are crying. Aww. Now, we aren't playing any sounds, and this is really important because looking at a baby do this is different from hearing the baby cry. Okay? But when a baby's pleasant or smiling, there isn't necessarily vocalization going on by that baby. It might be gurgling, but doesn't have to be. Okay, oh, I thought I took this out. Anyway, so we present them with a baby face. We present them with a face for 3,000 milliseconds, and then they rate how that baby face makes them feel. And then they get a distraction task, and then they get another baby face, and they rate it again. They get a distraction, and they're presented with 3 trials of baby faces, their own, another, smiling, crying. Okay? And we look at different brain areas. Here's the nucleus accumbens right here. Okay. And what Jennifer and Kat found was you get a greater bold response to own infant over another infant and to smiling faces over distressed faces in the nucleus accumbens as well as in the amygdala. So here's the bold response to positive, to negative. To positive in the amygdala, to negative. Here's the nucleus accumbens again. To own baby, to an unfamiliar baby. To own baby, to an unfamiliar baby. So and you see there are no left-right differences, by the way, in this case. We aren't getting any laterality here. We do get some laterality in cortex, but not in the subcortical regions. Okay, so basically, there's a greater brain activation response, if that's an adequate way of describing the bold response, to positive baby pictures over negative in the nucleus accumbens and the amygdala, and greater activation to own baby versus an unfamiliar baby. Okay. If you look at their self-report, it seems to sit similarly. This is now only in the magnet. There's more positive ratings of own baby positive than own baby crying. Not surprising. Okay. These behavioral and neurologic systems are set up by early neonatal and pre-weaning experience as main effects and an interaction with genetics in both the rat and in human mothers. And I'm just going to give you a teaser. And this teaser is because Viara Mileva, my graduate student, is right out there, and this is her PhD thesis, so she can describe it to you ad nauseum, and it's really a wonderful story. I'll just give you a little teaser. Okay. In a recent study, we related polymorphisms in dopamine and serotonin genes to maternal behavior and to maternal sensitivity in new mothers. We focused on the interaction between gene polymorphisms and aspects of early experience in the mother's family of origin. This is part of a big project that we're involved in with Michael Meany. You know Michael Meany? It's called the MAVEN Project, Maternal Adversity, Vulnerability, and Neurodevelopment. And it's a big project going on in Montreal as well as in Hamilton and in Toronto. It's a longitudinal study starting assessments right at the beginning of mother's pregnancy and following. The kids are now 5 years. Some of them are 5 years of age, and we're looking at school activity, etc. So we've been assessing the infant emotional, social, cognitive development, attentional development. We get genes and we get hormones, and I'm the mother end of this, and so I make sure that we get all of that from the moms as well. Okay. And this is just the teaser. The environment moderates the genetic effects on maternal behavior in 6-month postpartum mothers. What Viara did is she did many ways of assessing mothering. You can ask the mother how she feels by giving out standardized questionnaires that have been factor analyzed to assess feelings of nurturance or competence as a mother or self-esteem, etc. That's one thing you can do. You can do mother-infant interactions using a microanalytic procedure. You know, you can look at the amount of time the mother strokes the baby and talks to the baby and looks at the baby and looks away from the baby, and that becomes very important. Looking away from the baby. This is in a 20-minute observation. Okay, you can use the Ainsworth sensitivity scale to see how sensitive moms are to baby cues, how contingently she responds to their cries, so that when the baby cries, does the mother go, you know, is there a dance going on or not? And these are different ways of assessing. They do not necessarily correlate with each other. It's really interesting. It's the dissociations about what mothers say and what they do, which is really interesting, because they don't always do what they say. Okay, so this just simply shows that mothers, we also find out what kind of early childhood they had. So mothers can either have had a very positive early childhood experience. I'm sorry, this feels so loud to me. Okay, I can't. Okay, mothers either have a very more adverse early experience where there's been a lot of shifting in their early life, in the family of origin, a lot of shifting in who's parenting them, or not even shifting in who's parenting them. They can have the mom the entire time, but dad can leave and then come back, or stepfather can come in. Okay, that's called a consistent versus inconsistent care. But there are many measures that we use of that. In any case, what Viara found is that mothers with the GG genotype on the DRD1, dopamine receptor 1 marker, and it's called RS686, look away from their babies more frequently when they experience higher levels of adversity, but only if they're carrying this GG genotype. The ones carrying the other genotype show no differences. So it looks like those carrying this genotype are more responsive to the negative early environmental effects on this one measure of mothering. Now, we haven't looked at positive early experiences, and I think that's equally important. Okay. And you also see that mothers that have greater perceived stress and who are GG look away from their babies more, but not the other genotypes. Time. Sorry about that. Okay. All right, so I'm finished. I'm finished. Okay, so young are first attractive and then rewarding to new mothers as a result of both hormones and experience. Rewarding and salient young regulate mother's approach to and contact with the young. They enhance her motivation to mother. Attraction to young is mediated by the reward dopamine system, especially the mesolimbic system, activated by hormones, and the development of these brain systems are affected by previous experiences and by genetics. Thank you very much. These are all the people involved in my studies. Veronica, I just have to mention, Veronica Funso is a postdoc, and she did the dialysis study. Viara is sitting in the back there. She did the genetic study. Jennifer Barrett and Kathleen Wunsch did the imaging study, and other people did many other aspects of this research. Thank you.
Well, lunch is awaiting us downstairs. Shall we have one question?
It's a very nice study, and I wanted to ask your thoughts on the implications of your Fmri study. So I was very struck by the fact that that the mothers demonstrated a stronger signal to the infants when they demonstrated a more positive affect, so when the infants were happy. And it makes me wonder about the development of attachment. So when the way attachment is formed is when the mother is responsive to the infant, when the infant is distressed. And so the fact that the mothers showed a stronger responsiveness, at least on the Fmri study, when the infant demonstrated a positive affect versus negative affect, what are the implications in terms of maternal behavior for attachment development?
I would suggest the following, that we don't have an adequate stimulus for the negative side of this. That is, mothers are very used to hearing their babies cry. And that if we did videotaping or we did only auditory, we would see, I think, many parts of the brain become activated in response to the so-called negative stimulus. So I would suggest that the positive, we understand how that could enhance attachment. Okay? The negative part of this is how you would respond to an infant if you became very anxious and you wanted to bring the infant closer. So I think that in order to elicit that, we would, number one, need auditory. But number two, I think we would need to look at other brain areas as well. In particular, the amygdala is a place that I think is involved in a system of fear and of anxiety, and it's a very large structure. As it was mentioned, the central nucleus is very important for many of the negative features of fear and withdrawal and whatnot. So I also think that if we could do a more focused analysis of different parts of the amygdala, it's a very complicated big structure, that we would also see greater activation. We were very surprised. We were very surprised not to get greater activation in response to the negative stimulus.
Thank you.
Alison, one quickie. The mothers that were looking away that had adverse circumstances, could you interpret that differently as being used to an environment of risk where surveying the environment is more critical than looking at the baby? Rather than a sign of disinterest?
I think I would interpret that more in terms of her distractibility, her not being adequately locked in and moving if there are other people in the room, if there's other noise. She's much more readily, she will much more readily show with a move away response from the baby. Okay? Depending on, I think there are different reasons for looking away. It can be distraction. We do know that often mothers who have had a difficult early environment have attention problems. In fact, they show real problems in attention set shifting in particular. So that would feed into the idea that they're distractible, they tend to be more impulsive, etc. So I would put it into that framework.
Thank you very much. Our lunch is downstairs. We need to be back, though, at 1 o'clock for our next speakers. I'm sorry, we're a little tight here. And those who are speaking this afternoon, please bring your PowerPoints up.